Taste and smell. Thursday, May 14, 2020. 8:15 p.m. Taste and smell.
Olfaction structure and function. When you have a cold, you aren't able to taste things very well. When you eat, molecules travel up back of throat, and some go into back of your nose. So you're using your sense of smell in conjunction with taste. If your smell is knocked out, you can't taste things as well. Smell is also known as olfaction. Area in nostril called the olfactory epithelium. Separating the olfactory epithelium from the brain is the cribriform plate. Above the plate is an extension from the brain olfactory bulb, a bundle of nerves that sends little projections through cribriform plate into the olfactory epithelium, which branch off. At end of each connection are receptors, each sensitive to one type of molecule. Molecule travels into nose, binds one of receptors on nerve endings. Zoom in on olfactory bulb. Imagine there's olfactory cell sending projection to olfactory bulb. There are thousands of types of epithelial cells, each with diff receptor. Say this one is sensitive to benzene rings. When it binds to receptor, triggers events that cause cell to fire. AP will end up in olfactory bulb. All cells sensitive to benzene will fire to one olfactory bulb called a glomerulus. They then synapse on another cell known as a mitral/tufted cell that projects to the brain. The molecule binds to the GPCR receptor, G-protein dissociates and causes a cascade of events inside the cell. Binds to ion channel, which opens and triggers an AP. Pheromones. Why do dogs pee on fire hydrant? There are molecules released in the urine, which can be sensed by other animals through the nose pheromones. Their specialized olfactory cells cause some sort of response in animals smelling them. Pheromone is a chemical signal released by one member of the species and sensed by another species to trigger an innate response. Really important in animals, particularly insects linked to mating, fighting, and communication. Specialized part of olfactory epithelium in animals, the accessory olfactory epithelium. It sends projections to the accessory olfactory bulb. Within the accessory olfactory epithelium, you have the vomeronasal system. In vomeronasal system, there are basal cells and apical cells. They have receptors at tips. Triangle will come in and activate receptor on basal cell here. Basal cell sends axon through accessory olfactory bulb to glomerulus, which eventually goes to the amygdala. Amygdala is involved with emotion, aggression, mating, etc. Humans have vomeronasal organ, but no accessory olfactory bulb. Gustation structure and function. We have 5 main tastes, localized on the tongue: bitter, salty, sweet, sour, and umami, ability to taste glutamate. Taste buds are concentrated anteriorly on the tongue. Taste buds can be fungiform, anterior, foliate, side, and circumvallate, back. In each taste bud are the 5 receptor cells that can detect each taste. Each taste can be detected anywhere on the tongue. Mostly on anterior part of tongue. Each receptor has an axon, which all remain separate to the brain. And they all synapse on diff parts of the gustatory cortex. Known as the labeled lines model. X-glucose hits tongue, activates sweet cell, because it has sweet sensitive receptors, triggers cascade of events so cell depolarizes, and travels down axon to the brain. Glucose binds GPCR, conformational change, G-protein dissociates, opens ion channels, cause cell to depolarize and fire an AP. Sweet, umami, and bitter cells GPCR receptors. Sour and salty rely on ion channels. They bind to receptor directly, XNACL binds to receptor and causes ion channel to open, and plus ions outside flow in. Cell depolarizes and fires an AP. What happens if we put salty receptor inside a sweet cell? Receptors in membrane bind to glucose. But let's insert a salty receptor. Since axon from cell leads to brain, if NaCl comes in, it activates the receptor, plus ions go inside, sweet cell depolarizes and fires AP, and brain interprets it as a sweet signal.